Hi guys, in this video I am going to talk about hydrogen bonding. Now hydrogen bonding, if you compare it with coordinate bond, covalent bond or ionic bond, hydrogen bond is the weakest one. And it's very sensitive to temperature and pH. A bit higher temperature, hydrogen bond breaks. Or if pH very low, very high, hydrogen bond breaks. Alright, so it's very sensitive. But we cannot survive without hydrogen bonding. Now, if we take initially biological system. In biological system, whether it be our DNA, whether it be our cell membrane, or be it enzyme, hydrogen bonding is there. It's purely because of hydrogen bonding these faculties work. Alright, so to begin with, let's touch our catalyst that is present in our body, that's enzymes. Alright, so how is it important? Enzymes, we know, are very significant for us, for wear and tear of our cells. It's responsible for homeostasis. Alright, so now let's see how hydrogen bonding is involved there in catalysis or biocatalysis or enzymes. We have already seen in my earlier videos, enzymes takes primary fold and then secondary fold and tertiary fold, and since amino acids involved in enzymes are chiral, you would find they're all chiral, R up, H down, and so on and so forth, H up, R down, so all chiral molecules. Single enantiomer present there wherever there is amino acid, so it forms chiral pocket. So this is the chiral pocket, this is the chiral pocket, this is the chiral pocket as mentioned over here. So enzymes are highly enantioselective in their reactions. It reacts with one of the enantiomer, but with other enantiomer it won't react. If at all it reacts, it won't react the same way as it reacts with the counterpart. Alright, so this we have already seen in my earlier videos. Alright, now some more informations, data that I have given. Now mostly these amino acid enantiomers which are there, they are L enantiomers. Alright, in some mammals, D enantiomer of alanine, serine, aspartate, cysteine are also found. Alright, so basically if we check, it would be all L enantiomers, but in some cases we would also find D enantiomers. So okay, that's about enzyme. Now where does hydrogen bonding play? Why does this primary, secondary and tertiary fold occur? It occurs only because of presence of these hydrogen bonds that you would be checking out over here. Alright, so these are the hydrogen bonds which makes this polypeptide linkage to get into primary and secondary and tertiary fold and generate these chiral cavities, chiral pockets. Alright, so these hydrogen bond which we call as weak bond are responsible for all these enantioselective reactions that happens in our body or that we do in lab, calling it as biocatalysis. Alright, so again we find that weak hydrogen bonding taking care of enzymes. It's only because of this weak hydrogen bonding that enzyme works. Obviously, now seeing this, you know that it's very sensitive to temperature and heat. More of pH, it can break, or high pH, low pH, it may break. Temperature, these bonds can break, and once it breaks, then if it becomes linear instead of folds, then enzyme goes a waste. Alright, so that's the reason why we call it sensitive, but that's a different part of story. But all enzymes which are there, they are enantioselective, their activity is all owing to hydrogen bonding. Alright, so guys, wasn't it amazing, hydrogen bonding? Now let us get into another aspect, and that is about cell membrane. Alright, in case of cell membrane, because of hydrogen bonding, the cell membranes which are there, they are intact, and this is what protects ourselves from toxins. Let's understand this amazing aspect of weak hydrogen bonding also. This is a small bit of eukaryotic cell membrane, now which is also known as phospholipid bilayer. Now why is it called as phospholipid bilayer? Because it has got lipid part which is here, which is oil soluble, and it has got hydrophilic part, that is phosphohydrophilic part, which is over here as well as here. Now it is called as bilayer because you see that this becomes one layer and this again becomes another layer. Let us get into it in depth. Of the bilayer that I showed just now, I'm just choosing one layer, upper layer, right? Now upper layer, this is the head part of it, and this is the head part, or rather I would say this is the hydrophilic part and this is the lipophilic part. This is water soluble part and this is oil soluble part. Now let's understand why is it water soluble. Now if it has to be water soluble, then in this head part has to be polar, and let's check out whether it is polar or not. And regarding this, it has to be a fatty chain. All right, so okay, this is the chemical structure. Now here what I have chosen is phosphatidylethanol All right, now see, this is, if you check it out, it is ethanol, CH2O, and here you have got, instead of one hydrogen, that has been replaced by NH2. Now NH2 has got protonated, so it has become NH3 plus. All right, that is attached to phosphoric acid, phosphate group. All right, phosphatidyl phosphate group, and that phosphate is attached to glycerol part, and glycerol, in glycerol what we find is two of this alcohol. This is the primary alcohol, this is the secondary alcohol, which is in ester form, and it forms ester with fatty acid, could be lauric, linoleic, oleic, or stearic, palmitic. Now this CH2OH, it forms phosphate ester. There are two esters, all right, two long chain esters, and these are the lipophilic part. These are the lipophilic part. Whereas here what we saw, it was all polar. Why polar? Because this was NH3 plus, and phosphate of phosphate, this is also again negative. This is negative, this is positive. All right, now when we come down over here, we have got carbonyl groups. All right, carbonyl groups. So this is one head and tail. This is another head and tail. This is yet another head and tail, as shown over here. All right, now what we find is intermolecular hydrogen, all shown by red dotted, are intermolecular hydrogen bonds. So see, if we check out over here, this is the best example. Now if we take out there, now see, this is NH3 plus. All right, hydrogen bonded with this, hydrogen bonded with this. So this molecule is hydrogen bonded by there, and it is also hydrogen bonded by the other molecule over here. All right, see, with the other molecule over here, it would also be hydrogen bonded. Now let's understand the hydrogen bonding part. Now in case of hydrogen bonding, as I've shown here, now this head is hydrogen bonding here with this. Here it is hydrogen bonding with this molecule. Here it is, as a result of which they are all kept well tied with each other. Let's try to understand chemically. Now this was NH3 plus, so H hydrogen bonding with this oxygen. So this molecule is holding this molecule intact, and it's also attached over here. It's also getting hydrogen bonded over here. So it is keeping it securely tied with this molecule. Same thing we would be observing for all the molecules which are here. So that's what I've represented over here. So hydrogen bonded, hydrogen bonded. Now this is what is responsible for keeping the membrane intact, cell membrane intact. And mind you, we say hydrogen bond is weak. Now this weak hydrogen bond does the job of keeping all the toxins out of cell, and whatever desired material to be taken in is taken in a proper fashion. All right, so this is what is our intermolecular hydrogen bonding. And this phosphatidylethanol is second most abundant phospholipid mammalian cell membrane. All right, so again see how hydrogen bonding is responsible over here for keeping us safe. We know DNA holds the legacy of our traits or characteristics, and how hydrogen bonding is involved into it. All right, let's see and find out.
Hi guys, my name is Janvi and I am going to present the topic of hydrogen bonding in DNA. Hydrogen bonds are responsible for the double strands and properties of DNA. These bonds are between the two strands of DNA between the nucleotide base pair. With this hydrogen bonds involve the two nitrogenous bases, that is purines and pyrimidine, in which the nucleotide base pair ATGC, where A stands for adenine, T stands for thymine, G stands for guanine and C stands for cytosine. These bonds are of complementary bases are responsible to keep the double strands of DNA intact. So, the hydrogen bond of oxygen atom of carbonyl group and hydrogen of nitrogen keep the double strands of DNA intact.
Thanks, Janvi. So, what do we gather? We gather that these hydrogen bondings are very significant for us. Why? Because our traits or characteristics encoded in two strands of DNA are securely clenched by these weak hydrogen bonds. The legacy of these traits are endowed to incoming generations without fail meticulously when required. Bonds, though, are weak, but rarely fails to perform this critical responsibility with precision. Isn't it amazing? I hope you enjoyed this video on so-called weak hydrogen bonding. Still, there are lots that I intend to speak. I'll pick it up in my next video. Please wait for it. All right, see you then.